Hello and welcome to the webinar here at QPS today. My name is Sarah Cross. I'm the Executive Director and Head of Global Medical Affairs, and I'll be moderating this webinar. Our webinar today is focused on expedited programs for cell and gene therapies, and our presenter today is Dr. Kimberly Beitart-Hafen, who is the Executive Director and Head of Global Regulatory Affairs for QPS. As many of you already know, but if you don't, QPS is a GLP and GCP compliant clinical research organization, and we deliver the highest quality of services both across the preclinical space as well as clinical drug development services. We've been in business since 1995, and we've grown from a tiny bioanalysis shop to a full service CRO with over 1,200 employees. We have offices and services throughout across the globe, so based here in the United States, where both Kim and I sit, as well as offices in Europe and Asia. We offer expanded pharmaceutical contract R&D services, and we have expertise, as I mentioned, across preclinical and clinical, from neuropharmacology, DMPK, toxicology, bioanalysis, translational medicine, as well as running early phase and late phase clinical trials. We're an award-winning leader focused on bioanalytics and clinical trials, and we're known for our standards, our agility, our flexibility, and the approach that we bring to the work that we do in partnering with our clients. So we really are grateful for your time and to have you here for this webinar on cell and gene therapy. As a reminder, we also have another webinar in follow-up to this one, which you would have seen come through on your email, so feel free to register for that. That webinar is on excellence in complex clinical trials, and will take you through 3 case studies to illustrate the work that QPS has done in this space. So you can look for that. That is on Tuesday, October 18th. And just a reminder for everybody on here, this webinar is being recorded. You'll get an email with a link to the webinar, so you can always go back and watch this on demand afterwards. If you look over on your sidebar here, you have an opportunity to enter questions in, and we will collate those as we go through the webinar. So at the end of Kim's presentation, we'll go through a Q&A session. We'll answer as many questions as possible that you put into that chat box. Anything that we don't have time for or is unanswered, we'll certainly follow up with an email after the webinar. So with that, I would like to ask Kimberly to go ahead and take the floor, and we look forward to this presentation, Kim.
All right. Well, thank you, Sarah. Thank you for everyone that's joining us today as well. The topics that we will discuss today are gene and cellular therapies, safety and efficacy studies, industry challenges, global regulatory framework, and regulatory programs. If we start with an overview of gene and cell therapies, they hold an extraordinary potential to transform global healthcare. As a result, gene and cell therapies, the pipeline has grown tremendously. There's over 1,800 active and recruiting trials globally, and it's predicted by 2030 that more than 60 US approvals will be projected, and more than 500,000 patients are anticipated to be treated by these therapies. It all begins with the plasmid. Plasmids are utilized in gene and cell therapy. They're designed in a laboratory, so they're artificially designed, and they're designed to introduce foreign DNA into another cell. Due to their artificial nature, they're commonly called vectors or constructs. There are 2 types of vectors to deliver these plasmids to the cells. The first one is a viral vector. It's an inactivated viral virus, and it's used to be able to transport this plasmid through the cell membrane into the nucleus to be able to deliver that therapeutic gene. The second type is a non-viral vector. And if you think about our COVID vaccines, this utilizes the non-viral vector pathway. So with these, it can be lipid-based, polymer-based, peptide-based. It also allows that way of entering the cell wall and bringing that DNA information to the nucleus of the cell. There are different types of gene and cell therapies. If we talk about the first one, so gene therapy, it's the introduction, removal, or change in genetic material, DNA or RNA. Often a viral vector is used to deliver this information to the cells. There's a cell therapy, if you think about CAR T, where you remove the cells from the body. CAR T would be with apheresis, then you're going to transfect the cells with the plasmid, and then reintroduce these genetically modified cells. There's also gene editing, and the goal of gene editing is to remove, disrupt, or correct faulty elements of DNA within a gene. So this is really highly precise, and if you think of the CRISPR-Cas9 type of gene editing is what we are seeing today as a trend. There's different types of therapeutic indications for gene and cell therapies. You can see here that oncology is the #1 indication being pursued currently. Secondly, we see central nervous system, which is really an exciting time. A lot of CNS indications have not had successful treatments in the past. And then you can see there's very different, various types of disorders that are being pursued for gene and cell therapy treatments. If we look at the clinical trials, we're seeing that we've got a lot in the phase 2, which we'd like to see continue on to the phase 3, but the phase 1 pipeline is still going. So it's really, the industry is progressing in this manner through the different clinical trials. So we're seeing a lot of successes. I want to talk a little bit about precision medicine. So precision medicine uses next generation sequencing. So you can rapidly identify or sequence large sections of a person's genome. And you're looking for genetic variants that assist with the diagnosis and treatment of human disease. Pharmacogenomics is the study of how genes affect a person's response to drugs. So in this area, you combine pharmacology, the science of drugs, with genomics, the study of genes and their functions, to develop effective, safe medications and doses that are tailored to variations in a person's genome. A little bit about rare and, here we have the diversity of these products. So you can see the starting material. Is it going to be cells or tissues? Where are you going to derive these materials from? Are they going to come from the patient or a donor? Are we going to start with stem cells, which can be any type? It can transfer into any type of cell in the body. Is it going to come from an animal, bacteria? And then there's vectors. We talked about the viral, non-viral. There's also bacterial vector. And then how are you going to integrate it into the host cell? Manipulation, various forms of manipulation. So, you know, where are you going to transduct the cells? How are you going to select the cells? How are you going to grow the cells? And then how are you going to harvest that product material? Often you need to purify what you've harvested. So column purification, filtration, concentration, and then what's the final formulation? Is it going to be frozen? Will there be carrier cells? Is it going to be lyophilized? So these are all things that need to be considered, and you can see how variable these products are. So they're all very unique, and that's why we have that unique regulatory framework. So there are challenges for cell therapy products. So here we're seeing stem cells, which can become any cell in the body. So here we have perhaps cardiac, skin, liver, and it's all about the kinds of cues that you give them and which directs them down different pathways. But the concerns are you going to have tumor or ectopic tissue formation? Is there going to be toxicity or mechanical failure? Often cells are transplanted into the body on some type of scaffold, And the typical platform for that would be to have it degrade over time so the cells can become used to their environment. But you need to take into consideration any kind of impact the scaffolds would have on the patient. Then there's unknown donor or cell fate. Is there going to be survival or persistence of the cell? What's the phenotype, distribution following administration? So this really needs to be evaluated as part of the preclinical testing program. With cell therapy, you have autologous versus allogeneic. So with CAR T today, we see the autologous model where you have one person donate their cells. The cells are manufactured and then brought back to this one patient. What's in clinical trials and what is the future is the allogeneic, so the universal donor, the off-the-shelf. So you have a patient that donates. In manufacturing, they're going to expand those cells, and then you can treat many, many patients with one person's donation. So how do you go about doing this? Well, there is this human leukocyte antigen complex that's on all cells. And this is how the body recognizes foreign from themselves. So in your typical donation of cells, say bone marrow transplant, you have, you're going to look through a large population of donors and see who has that HLA match, and then you match the donor to the patient. And this has been a problem over time, getting that match so that you don't have graft versus host disease, where the body attacks the cells. So a solution is, and we talked about that, gene editing, using perhaps CRISPR-Cas9 to remove this antigen complex, and then you have that universal donor where you can donate these cells to different patients. So let's talk about the gene therapy product challenges. And really, we want to emphasize that preclinical testing program. We want to make sure it's safe before you go into your clinical trials. So you want to assess for adverse immune responses to the vector or expressed transgene, the level of viral replication in non-target cells and tissues, insertional mutagenesis or oncogenicity, viral distribution, and transgene expression. A target product profile is a really great way to set yourself up for success. You start in the pre-clinical stages and you're considering your overall clinical program in the design of your target product profile. It facilitates discussions between the sponsor and regulatory agencies, and it's going to plan out your entire developmental process. You want to keep in mind what you want your labeling claims to be, and you want to build the support of that into this target product profile. I'm going to talk a little bit about orphan and rare diseases. A lot of gene and cell therapies are developed for these type of diseases. Often they'll have one mutation in their DNA, and that can be targeted and that can be corrected. So, but what are the implications of this? We have small patient populations. You need alternative trial designs and alternative statistical techniques. You want to maximize data from that small population and its potentially heterogeneous groups of subjects. And we're seeing a lot more single arm designs with historical controls. And next we'll talk about safety and efficacy. It really starts with your evaluation of the preclinical data. You want to get information from your preclinical data that gets design or guides the design of the clinical trial. For example, the identification of no observed adverse effect. What level is that at? Additional animal studies may be necessary during late phase development after clinical trials have been initiated, and they're often conducted at the same time as the phase 3 trials. Examples of these would be developmental assessments and reproductive toxicity. The clinical trials, things that you want to consider, we're looking at rare diseases, so large trials are not feasible. And we're seeing early and compelling signs of clinical efficacy, resulting in accelerated programs. So that typical paradigm that we're used to with the phase 1, 2, and 3 is now getting, the lines are getting blurred and moving faster. So we have that phase 1, 2, because we're getting that early efficacy, and then the phase 3, but the post-approval studies are becoming very important because we want to gather that long-term data, and we have really moved quickly through the early phases of clinical studies. And so regulatory agencies are just more open to discussions about innovative designs and for these clinical trials. So early phase studies are becoming very, very important because you're enrolling subjects that have the disease or medical conditions. It's not ethical to alter the DNA of healthy volunteers. So you still have that primary objective of the safety evaluation, but also we can add on looking for evidence of bioactivity, of characteristics of the disease. A single administration dosing regimen, often it's a one-time dose for a cure, which is just really, really exciting. But we're not going to have a lot of that early safety data that we're used to. So you want to be careful when you're looking at your different cohorts by staggering, not doing a whole cohort at once, just allowing for that time to assess for any types of adverse events. There are many industry challenges involved in these types of therapies. We talk a little bit about them, product characterization and testing challenges. There are a few industry standards and reference materials. Often we're looking, if we're talking about orphan indications, it's small scale. We don't need to have large, like in biotechnology with bioreactors, we're not looking at that type of scale. There's a lot of patient-specific lots. If we're talking about CAR T, you're really manufacturing that for the patient. So there's a lot of lot-to-lot heterogeneity, limited shelf life and stability information. And then these products are highly specific. So the strategies for testing and storage will be often unique to the product. There's challenges in development. Establishing the quality, safety, and efficacy data to support that benefit risk profile. Challenges include, finding that correct dose estimation, the development of manufacturing processes and associated quality standards. Invasive routes of administration, you really want to get the product to where it needs to be and avoid a lot of the having it go in different parts of the body. So that can be a challenge. We're seeing a lot of catheter administrations. The first gene therapy product was for an eye disease. So that's one of the easiest routes of administration. You got that small patient populations, and there's a lack of established clinical endpoints. So, that gives you, there's some flexibility in design. And then the long-term risks, and that's why we want to look at those post-approval studies. You want to assess for immunogenicity, the ability to form tumors, and is there any chance for the loss of expression over time? There are supply chain challenges. Raw materials often are derived from human or animal materials, which has a lot of biological complexity. There's variability of lot-to-lot performance characteristics. In our traditional GMP environment, we have that highly controlled. So kind of moving to a different, more flexible manufacturing environment. A lot of these materials are only exist in research use only, and agencies are really looking to have that paradigm shift. You want to have as much quality materials as you can, especially because you're going into the human a lot faster than the traditional pharmaceuticals. And we are talking about biologics, so the import and export requirements can be difficult for those starting materials, clinical samples as well, and then the finished product. There are manufacturing challenges. So this is your typical quality by design, where you take your clinical outcome, your what you want your clinical outcomes to be, and then you trace them back to your critical quality attributes to have that full characterization of the quality by design. But here with gene and cell therapies, the critical quality attributes are not well established. So demonstrating that link with the clinical outcomes is difficult. And then we're seeing that early efficacy. So we're going to commercialization faster. And what happens is your CMC, your chemistry, manufacturing controls. So it's difficult for them to keep up. They really need to get to full GMP by the time you go to commercial, and it's happening faster than your traditional pharmaceuticals. Additional manufacturing challenges. So there's a high cost of goods, long lead times, significant upfront investments. There's limitations on these viral vectors. There's just an increased need for them because there's increased number of therapies being developed. And then we are seeing increase in the size of populations to be treated, which is very exciting. But there's a lot of use of contract manufacturers, so there's long wait times to get your clinical trial materials, and then there's also long wait times to be able to manufacture them in these contract manufacturers because of the high demand. So there's global challenges. The key challenges here are really getting the regulators to align. You want to, especially with the rare diseases, you want to maximize the information that you get for your clinical trials. So you really want to work to have them align, but we are seeing a lot of differences in novel or surrogate endpoints requested by regulatories to assess changes in gene or protein expression. There's differences in requirements for study duration, timelines, documentation. For example, environmental risk assessments and genetically modified organisms applications, they vary between each member state. Another important thing that we're seeing here is that post-market confirmatory studies. So safety and efficacy data, it's very limited by the time we're pushing these through to commercialization. So it's very important to have those long-term patient follow-up studies and to develop disease registries to get that efficacy and safety data for the risk benefit profile. So now we'll talk about the global regulatory framework. So some countries have established guidelines and frameworks where they do have the experts in the field that can help you with your developmental pathway, but there's a lot of countries that just don't have the structure or the scientific knowledge to be able to do that. So sometimes they group them with biologics, but they really are unique. And we did talk about the complexity. So Unfortunately, countries that don't have all of these specific scientific advice and specific areas for guidance, they leave patients without the treatment that they need. But we are moving towards global regulatory harmonization. Some examples of this are confidentiality commitments and memorandums of understandings, where regulatory agencies can confidentially share information with other regulatory authorities. An example is the parallel scientific advice. It's an example of this where the FDA and the EMA share information and offer joint advice for a specific product and indication. So the interactions can provide information on about scientific advice. It's an opportunity to optimize product development and avoid unnecessary replication of testing or divergence in testing methodologies. Clusters are another example. There's a forum in which the FDA and other regulatory authorities discuss specific areas of mutual interest. For example, the Advanced Therapy Medicinal Product Cluster is specific for gene and cell therapies. There's other international forums. For example, International Pharmaceutical Regulators Forum for Cell and Gene Therapy Working Group and a Gene Therapy Working Group. It's an opportunity to share scientific knowledge and regulatory experience. Other regional initiatives include Pan America Health Organization and the Asia Pacific Economic Cooperation Harmonization Center. And the interests are in the convergence of regulatory approaches for gene and cellular therapy. There is an initiative for standard development. The FDA is looking at these activities, including participation in initiatives to develop international standards with the goal of harmonizing regulatory expectations internationally. For example, gene and cell therapies that use the same vector platform, they could be compared in a similar manner for how they should be manufactured and how they should be tested and how they should be studied preclinically and clinically. And this will facilitate product design and reduce product time to market. The global regulatory framework is which regulators experienced in gene and cell therapies have adopted requirement practices that are unique to the development of these products. So the EMA and the US FDA have been very forward-thinking in this by developing guidelines and guidance documents specific to these types of products. And we're also seeing similar initiatives in Brazil and Canada. There are established regulatory frameworks. We see this in the US, the EU, and Japan. So in the US, we have the US FDA, the Center of Biologics Evaluation and Research. There's an Office of Tissues and Advanced Therapies. In the EU, in addition to the European Medicines Agency, there's the Committee for Medicinal Products for Human Use, and they specialize in advanced therapies. That covers the gene and cell therapies. And in Japan, the Pharmaceuticals and Medicinal Device Agency and the Ministry of Health, Labor, and Welfare has the Office of Cellular and Tissue-Based Products. So we'll talk a little bit about regulatory programs, including expedited programs, priority review, and accelerated approval. So expedited programs, really focusing on that pre-submission phase and looking at increased collaboration with regulatory agencies prior to submission of that first in-human dossier. With these programs, you can cut at least 2 years off new drug development. They're not going to change the standards for approval, but they will change that risk-benefit ratio. So if you're seeing that high benefit in early stages, there's more an acceptance of risk. The sponsor still has to do all the required studies, but we may see more post-marketing commitments to show that that early data did show up. We're seeing a lot more collaborating with regulatory agencies, and adaptive R&D process. We're moving to that phase 3 much more quickly than a typical pharmaceutical development timeline. And there are strategies that are really important to consider early. So your regulatory, your clinical development, your commercial strategy. We're looking at shortened developmental timelines with these pathways. They provide significant advance for products that provide significant advances over current treatments. Or maybe they are the only option for serious diseases. So it's a really exciting time to offer treatments, perhaps a one-time cure for patients that had no treatments prior. And the goal is just to get the patients treatment as fast as possible. So expedited pathways include designated programs that offer mechanisms for increased early communication with regulatory agencies to facilitate that streamlined process. Examples of these are you can use your preclinical data to apply for fast track, and this is in the US. If you have some early clinical data, you can apply for PRIME, that's the EU, or Sakigake in Japan. You have increased access and feedback from regulatory authorities. They're ones that are actually going to grant the designation. And through frequent meetings, the sponsor and regulatory authorities can achieve alignment on study designs and data requirements. Priority review and accelerated approval. So priority review and accelerated assessment programs provide shortened review timelines for marketing authorizations. So now we've moved to we have our clinical data, and we're putting together the application for the marketing authorization. There are opportunities for accelerated approval and adaptive licensing, such as conditional approval. For this, you see more of the use of surrogate endpoints and authorization based on non-confirmatory evidence, but this still needs to be assessed after marketing authorization. We talk about the priority review. You can see here we have the US, the EU, and Japan. In the US, with priority review, you cut that timeline from 12 months down to 8 months. In the EU, from 210 days to 150 days. And in Japan, 12 months down to 9 months. Accelerated approval. So the US has accelerated approval. The EU, it's called conditional marketing authorization. And Japan, it's called conditional and term limited approval. So it's possible to only need to conduct the first of 2 pivotal trials to use a surrogate endpoint as the efficacy endpoint for a pivotal study to achieve conditional approval. The key component of any expedited registration pathway is that confirmatory studies need to be conducted after the conditional approval in a timely manner. So there's also situations where marketing authorizations can be granted without confirmatory studies. It's really an ethical decision. If you're not seeing signs of issues with toxicity or any kind of adverse events early on with your preclinical and your clinical data, and you're seeing that efficacy, the potential for cure, we really want to get it to the patients faster. So taking on that risk, but also committing to the post-approval studies and the generation of disease registries. So in the United States landscape, These products are classified as human cells, tissues, and cellular and tissue-based products. They're regulated under the Centers for Biologics Evaluation and Research. There's 4 expedited programs: Fast Track designation, Breakthrough Therapy designation, Accelerated Approval, and Priority Review designation. In the EU, these products are defined as advanced therapy medicinal products. They are regulated through a centralized marketing authorization. There's 2 committees that review and approve the clinical trial applications. That's the Committee for Advanced Therapies and the Committee for Medicinal Products for Human Use. The sponsor must submit post-marketing confirmatory studies before that conditional marketing authorization can be converted to a standard marketing authorization. There are exceptional circumstances where the marketing authorization is granted without comprehensive data when it's not possible due to disease rarity or unethical considerations. In Japan, these products are classified as regenerative medicines. They're regulated by the Ministry of Health, Labor, and Welfare. They're unique. They offer unique fast-track approval frameworks. There's conditional timeline marketing authorization that can be issued based on preliminary clinical trials, indicating efficacy and confirmed safety. If they get that conditional marketing approval, they have a maximum of 7 years where they need to update during that time and have the post-marketing evidence submitted to the agency. So in conclusion, there's a rapid expansion of these therapies that offer the potential to treat diseases with unmet medical needs. There's a unique nature of these therapies that pose challenges, and that's why you want to really work with regulatory agencies, and now we have ones that are developed to specifically work in these areas and really help these products get to the market. So regulators have established new dedicated frameworks, and the other countries that will be moving down these pathways need to consider critical elements when developing these regulatory pathways, such as early engagement with regulators, innovative clinical trial designs, and post-market confirmatory studies. So increasing the alignment of regulatory pathways across countries is critical to facilitating the development of and access to gene and cellular therapy products on a global scale. Thank you very much.
Kimberly, thank you so much for the informative presentation on expedited programs in cell and gene therapy. As you were speaking there, we had a couple of questions come in, so maybe we'll move to the Q&A section of this webinar. You know, I think from your regulatory point of view, help us understand how the pandemic and Project Warp Speed has impacted the regulatory landscape.
Okay. Well, the pandemic was not a positive thing for most people. And we did work through it to the other side, but something really amazing that happened was, you know, regulatory agencies coming together, working with companies, and really getting the vaccines to the market in 2 years. It's unprecedented. And how they did it was they really used platform technology. So what type of vector are you using? What they put together the standards for it. They offered financial support, and it really was everyone working together, companies working with regulatory agencies, getting it as fast as they can to the patients. And we also brought on new technologies. The mRNA technology, it had not been commercial before. And what we're seeing now is additional indications. So if we can use what we learned getting through that regulatory fast pathway expedited to push these technologies through faster, we're going to get to more of those orphan and rare diseases and get the treatments to the patient. So a positive that came out of it was really facilitating products to the market, and that was a really exciting initiative.
Yeah, thank you for that, Kimberly. I think echo the same sentiment. The silver lining really was, I think it helped us rethink a lot of the processes that we have in place, and how do we, challenge ourselves to think differently and speed up some of these important pieces of the puzzle as it relates to the asset development program. The next question I have here for you is really about what's the best strategy for global approval of a product with a rare disease indication?
Okay. Yeah. So, rare diseases typically are not confined to one country. And if we go, patient-centric, we really want to roll out technologies to treat these rare diseases globally. Ethically, that's what we want to do. And so it's really great to see these initiatives where different countries are having some forums and trying to align on different pathways to facilitate to the patients. So really want to tap into those. Any kind of joint feedback we can get from regulatory agencies is really going to facilitate that pathway to the patients. So I recommend having a global view of your clinical studies, and also aligning and using some of these initiatives to have the agencies come to the similar requirements for your clinical and confirmatory studies. So you want to do that early on, have these cross-functional meetings with regulatory agencies globally.
Very helpful, Kim. And we have another one here, which is, maybe from your point of view, are you able to indicate what a typical non-clinical tox package might look like to support a first-in-human study investigating a gene therapy product?
Okay. You have all of your traditional pharmacology, PK, DMPK, and toxicology, but something that's really unique about the gene therapy and the preclinical is assessing for immunogenicity, assessing for that distribution. Is it going to hit your target, or are you going to have any kind of distribution of the product that impacts? So it's really important to have that understanding prior to going to humans. So there is more preclinical work in place to understand, is it going to have any off-target issues? Is the body going to react to it? And are you getting it to where you need it to go? So you really want to think about it scientifically, and then bring your plan to regulatory agencies, because as we learn more, there is more requirements. So really want to be up to date and to align, especially if you have something that's novel with the gene therapy, a new platform technology, then you want to align on these with the regulatory agencies.
Super. And I know our teams have been involved a lot on this type of work for clients. So something that that you have near and dear to your heart. And I think as an organization, we have that full service capabilities to provide that support for clients. So really appreciate you sharing that, Kimberly. I think, maybe one additional question is, how does a regulatory agency assess a virtual company for regulatory compliance?
Okay. We're definitely seeing more and more of this. Really the skeleton in the beginning, just to make sure They build the company and make sure the product's going to go through. So we see a lot of virtual, definitely even more coming out of COVID, where a company doesn't even sit together, and they just have the the scientists, the quality. They often don't have regulatory, and they're getting the investors. So what's really important in that is quality agreements, because they contract everything out. So quality agreements and the Toro, you want to clearly define who is responsible for what. Everything needs to be in writing. If it's not in writing, it doesn't exist. But ultimately, the sponsor is responsible. So yes, you delegated GMP to your contract manufacturer. Your contract manufacturer manufactured with poor GMPs. You are responsible. It's your product. You are the sponsor. So while you have these contracts in writing, you are responsible for the product. And that's something that I have to tell clients often. Yeah, so it's just more and more of that, making sure you have that quality oversight of the manufacturing of your product and the testing of your product clinically.
Fantastic. Well, with that, Kimberly, I think we'll close out the Q&A session. Again, thank you for the highly informative presentation today, and thanks the participants, all of you, for your time. We cannot thank you enough for taking the time to listen and ask your questions. I think just a couple reminders for the audience: if there are outstanding questions, please know we will get to those and follow up with an email. Also, you'll have an on-demand link that will be sent to you with this webinar if you want to go back and revisit anything else. You can also certainly visit qps.com to download a white paper that is on this same topic for additional information, and you can reach out and contact QPS through our website should you have any follow-up questions. And lastly, I'll just remind those of you that are on the webinar that we have a follow-up webinar, Excellence in Complex Clinical Trials. We'll review 3 pretty unique case studies and how QPS has successfully been involved in some interesting studies, and I would say, really delivered on the needs for patients. If you've not done so, go ahead and register. It's in the same email that you got regarding this particular webinar, and that will be on Tuesday, October 18th at 10 a.m. Eastern Time. Again, with that, thank you all for attending. Really appreciate the time. And Kimberly, thank you for taking us through this informative webinar.
Thank you.